Good afternoon. It's a pleasure to be here, presenting on behalf of UniQure. Thank you for the invitation. What I'd like to do today is spend a bit of time talking about an overview of UniQure as a company, and then go a little more into detail about some of the key programs that we're moving along. And this, of course, says that these are forward-looking statements which are inherently uncertain. So we like to think of ourselves as we're a leading fully integrated gene therapy company. This word is important to us, fully integrated. When I first joined the company, we were speaking so much about Glybera. We were the Glybera company. To some extent, we still are. And it was all about platform and technology. Clearly, it's still very important to us, but we think that we're moving away from that with a number of key achievements recently. We announced about a month or so ago positive phase 1/2 data in Sanfilippo B, which we believe validates our AAV5 in the brain. Unfortunately, when we announced this, there was a big mess going on with our friend Martin Shkreli, who raised the price 5,000% of an old generic drug, and Hillary Clinton's response. And right in the middle of all that, we made this announcement. So it went by a little bit unnoticed, but we think that it's great, and I'll show you why in a little bit when I get to the data. We hope to have very shortly near-term results in a phase 1/2 study in Hemophilia B. We earlier this year had a $2.3 billion partnership for cardiovascular gene therapies with BMS. This is interesting, and this confirms our desire to be a vertically integrated company as we move beyond technology platforms and into driving drugs to patients. We have reorganized our company into 3 distinct therapeutic areas. We have TA leads for each one. We're moving more into a matrix type of organization. We're starting to see some of the problems that companies much bigger than us are dealing with, but we're working through the growing pains, and this is a direction that we're going on in the future. We are very proud of our industrial scale insect cell-based manufacturing platform. And we continue to refine and to work with partners to accelerate the development on vectors and look for new synthetic, smaller promoters that we can put into our capsids. And we have ample capital to execute on all of this. So just a word on our manufacturing. We began in Amsterdam. As you can see, the scale was rather small, 2 by 50 liters. This is where we began producing Glybera. This is an EMA-approved facility, but to meet the needs going forward, we began investing about 2 years ago into a state-of-the-art facility in Lexington, Mass, which we believe is the world's largest gene therapy manufacturing facility, 50,000 square feet. We're working towards initially 2 by 500 liters, but this is scalable to 2 by 2,000 liters. And so as we've done the analysis internally of all the products which we think could be coming in our pipeline, we have ample room to meet all of these needs. And we're looking to get GMP validation in the next couple of weeks. This is what makes the scaling up possible as proprietary insect cell-based technology. It's safe, it's highly scalable, as you've seen just above, and it's cost efficient. And the basis is the baculum-based manufacturing, which we believe is the future in gene therapy manufacturing. Now, I mentioned we also continue working on our vector design and the promoters. I won't go into all of that, but you can see that we have a number of alliances, and we're not content to sit back and use what we have already had. We continue to revise, to improve, and to make the technology even better. So I mentioned earlier that we have split into 3 distinct therapeutic areas, and so you can see that over here. So the first one is liver metabolic, and of course Glybera is up here. Now the blue is products that we have partnered, and orange is things that we still have ourselves for now. And my friend Diego spoke this morning a bit about Glybera. We retain the rights for US, Canada, and other countries. Hemophilia B is also partnered with Chiesi, but again, we retain the rights for US, Canada, etc. The next area is CNS, and here we have the data which I mentioned to you earlier, and I'll show you in a minute about Sanfilippo B. We're also working on Parkinson's, and we've just recently taken the decision to move our Huntington's program from from development into research. And finally is the cardiovascular therapy area, and this is based on the partnership that we have with BMS, which we concluded earlier this year. The lead program is S100A1, and I'll mention a little bit about that in a bit. What's also interesting here is what we call our farm team. So we have multiple preclinical programs in all of these areas, and these are programs which don't require a lot of funding at this point in time, but we have the capability to move these into clinic if and when, as soon as we have interesting data for all of these, and there are quite a few. So first, a word on Glybera. The indication is orphan, actually ultra-orphan disease, LPLD. There was no other therapy approved before Glybera. It is very rare, as I said, ultra-orphan, so 1 to 2 per million. So if you look at the total LPLD population, it's about 500 to 1,000 in the EU. The label, if you then apply the label to that 500 to 1,000, you end up with quite a few less patients than that. So you remove the non-adults, you remove those who are controlled by diet, you remove those who have not had multiple or severe pancreatitis, and so you end up with a relatively small number of patients. But that's okay, because for these patients, we have data which currently shows 50% reduction in pancreatitis for these patients up to 5.8 years, as Diego mentioned this morning. And we're very proud to report that as of September, so last month, the first patient was treated with Glybera as a commercially available, reimbursed gene therapy. Now, it's not a national reimbursement. We're still going through those, as actually Chiesi is still going through those, as Diego mentioned this morning. But we have received reimbursement. In some countries, you're able to do negotiations between a hospital and a sick fund on a single case basis, and that's what happened with this first patient. So we have regulatory, we have commercial validation of both our AAV technology and our insect cell-based manufacturing. And again, moving away from being the Glybera company, we leave this now in the very capable hands of Chiesi, and we believe that the future value of UniQure is based on what you're going to see now. So, first, Sanfilippo B. It's a devastating, horrible disease. It strikes children at a very, very young age. It's very rare. Thanks for the kids. Autosomal recessive disease. The missing enzyme is NAGLU, and what happens is you get a buildup of toxic metabolites, GAGs, glycoaminoglycans. And then what we see with the children as of 2, 3, 4 years old is a progressive deterioration in cognitive and behavioral function. So unfortunately, this leads to death. The average age of death of this disease is about 17 years old. So it's just a horrible, devastating disease. There is a significant, as you can imagine, unmet medical need. There is no drug that currently works, no treatment option approved for these patients. We believe there are a couple of hundred. It's a very poorly known disease as well, because there are so few patients. So there are no, we know where the patients are, but there are no statistics that are predominantly available. So we believe there are a couple of hundred in Europe, a couple of hundred in the US, up to 2,000 if you add Canada, Australia, Brazil, Japan, etc. So the study that we are reporting results on here is a phase 1/2 study. It was started in October 2013. It has 4 patients. It's a small study, but again, these patients are extremely rare to find, and we wanted to find patients that had just begun the cognitive deterioration to see if the drug can bring any benefit. So all children had 0% NAGLU expression at the baseline, and we followed them up for 1 year at 1, 3, and 12 months time points. The treatment is administered directly into the brain. There are 8 burr holes, and each burr hole has 2 different depths, so there are 16 administrations. The findings were terrific. No safety concerns. We have robust biomarker results, so restoration of NAGLU from 0 to 14 to 17% of normal, and an effect that was persistent at 12 months. So these are what we consider extremely encouraging signs of efficacy. So the conclusions are that you see the NAGLU is restored. Even a couple of percent can help these patients to continue or to slow down the cognitive deterioration that they've been experiencing. It's safe and well tolerated. Again, we believe that it validates our AAV5 platform, and we will be showing 30-month follow-up data by the end of 2016. The next program I'd like to mention is hemophilia. Now we're going from a ultra-rare to a rare disease, and I think probably everybody knows someone who has, or someone who knows someone who has a form of hemophilia. It is still nevertheless rare. There are about 28,000 hemophilia B patients worldwide. This disease, compared to Sanfilippo, is very well known. There are registries or databases. We know exactly where each patient is. So this is due to a dysfunctional factor 8 or factor 9. And again, the medical need, there are treatments, but the treatments are just not that easy to take. It's frequent, 3 times a week, infusions, painful, time-consuming, and compliance is very challenging. So what we see here is, like I said, a couple of times a week, and what these factor infusions will do is restore levels, but only for a couple of days, the half-life being extremely short. And if you are at less than 1% of factor expression, you're considered a severe patient. Up to 5 is moderate, and then these are the mild patients. So somewhere before you get your next infusion, you are moving towards a severe phenotype again with risk of bleeding. So the idea is that if we can give gene therapy and the endogenous factor 9 remains above 5%, then we're keeping these patients away from these spontaneous bleeds. Now, we started a phase 1-2 study. Here we have the doses. What's important about this slide, here we go, is that we cross-referenced 2 previous studies, a study from St. Jude's, which looked at the safety, but also some of the response rates of factor expression, and then a porphyria study with our vector. So the St. Jude study was not our vector, but it was our gene cassette, and this one was our vector. And by cross-referencing these, we were able to immediately start with what we believe a therapeutic dose, and then a higher dose in our second cohort. So I'll let you read all of that, but what's important to know is that we have actually begun treating cohort 1 a couple of months ago. We are pretty much finished with cohort 1. We should be reporting in a couple of weeks on the interim analysis, and we will begin cohort 2 early next year. All right, the BMS partnership. This is again in the cardiovascular space. We have multiple cardiovascular programs. It was a well-publicized partnership a couple of months ago, at the beginning of the year, so I think you're all familiar with the terms. But what's most interesting here is S100A1, which is the lead target, which should enter the clinic in 2017. And just one slide to show why we were so excited, and presumably what excited BMS as well, is a 1-year survival in, and as we heard earlier this morning, the pig model is the best approximation of the human heart model that we have. And here we have 1-year survival data, so compared to, a S100A1 versus not, and 90% survival, pretty close to the sham control group. We also have biomarker results. So here you have troponin T and BMP. The top one is BNP, and this is a amino acid peptide which is secreted by the ventricular myocardium in response to strain. And so you can see that with S100A1, it is really brought under control, which means presumably that the strain is controlled. And here with TNT, the same type of effect when you have S100A1. I'm running out of time, so I shall wrap it up very quickly. I think this is just a summary of what I went through, but more importantly is where we're going. So the idea is we really do look to the future. We really look to one shot and done. We've done Glybera. We have data in Sanfilippo. We have hemophilia data coming up. I mentioned what's important to us is if we move to the next step of becoming a fully integrated gene therapy company. We hope to bring 2 marketed drugs in 2020. So we believe we have the leading portfolio, and we continue to progress to bring these therapies to patients. Thank you very much.